Cytology, biological membranes, cytoplasm. The main questions of the lecture. Plan of cell structure. The cell consists of cytoplasm and nucleus. It's separated from environment by cell membrane. Cytoplasm includes heloplasm, where suspended organelles and inclusions. The cell is divided into compartments by biological membranes. The structure of universal biological membranes. It consists of bilayer of lipids, where phospholipid molecules directed their polar hydrophile heads outside, and their hydrophobic tails into inside. In this lipid sea, whereas proteins can move, swim in lipid bilayer. There are surface proteins on the surface of the membrane, transmembrane and outer membrane proteins. This is a liquid mosaic model of biological membrane. If you use the freeze fraction method and remove 1/2 of lipid bilayer, one layer of phospholipids, like here, and look at that through the scan electron microscope, you can see the protein globules. Universal biological membrane thickness between 6 and 7 nanometers. The cell membrane or plasma membrane is the thickest biological membrane of the cell. It contains a universal biological membrane in the middle and has glycocalyx at the external surface. Glycocalyx consists of carbohydrate chains connected with lipids or proteins, and it produces complex glycolipids and glycoproteins of cell membrane. Glycocalyx responsible to cell recognition and adhesion. Under biological membrane, from the cytoplasmic surface, there is submembrane layer, underlying part of cytoskeleton. It consists of microfilaments, usually actin microfilaments, and microtubules. This submembrane layer provides the shape of cell and its movement as well as exocytosis and endocytosis. This is a nice picture of cell membrane. You see the lipid bilayer and protein molecules embedded inside. You see the glycocalyx outside. And types of proteins: structural, support the structure of cell membrane; transport, provides the transport of different substances; enzyme, catalyze the chemical reactions; and receptors, like biosensors, accept chemical or physical signals from outside the cell. Functions of membrane: demarcation, separate cell from environment; recognition of other cells; intercellular communications; transport of substances; interaction with signal molecules; and participation in cell movement. Receptors of a cell membrane: biosensors. It's the complex protein compounds. They coupled with G proteins, adenylate cyclase, guanylate cyclase, and phospholipase C, and some other enzymes, which generate second messengers like cyclic adenosine monophosphate, cyclic guanosine monophosphate, phosphoinositol, or calcium ions. They are second messengers, intercellular messengers, which mediate the effect of signaling molecules which affect the receptors. Transport through the cell membrane. There are two types of transport: by simple diffusion. The small molecules can go through the cell membrane, like water, oxygen, carbon dioxide. From the region this high concentration to the region of small concentration. On the concentration gradient, it not require the energy, and facilitated diffusion, which use ion canals or protein transporters. Another group of transport processes named active transport. It requires energy because the substances going against the concentration gradient and require the energy of adenosine 3-phosphate. The large molecules and particles pass in the membrane by exocytosis outside cell during the secretion process, and endocytosis. Then substances, particles go inside the cell. There are two types of endocytosis: phagocytosis for bacteria and dense particles. You see the leukocyte, which using pseudopodia catch the bacteria and enter it inside cell covered by membrane, and then digest using the lysosome enzymes. And pinocytosis is the transport of fluid, droplets of fluid, with diluted substances. And germinal cells normally have a gap about 20 nanometers between cell membranes. Now, there are two types of intercellular junctions: unspecialized or simple, and specialized. Simple junctions, then cells closely connected and connected but the glycocalyx molecules. There is also the junction and then extension of one cells enter the invaginations of neighboring cell. Specialized junctions may be of three types: anchoring junctions, like desmosomes or adhesive spots or macular adherens. Then transmembrane proteins of two cells connected strongly, and they are connected but macula densa, which connected these intermediate filaments of cytoskeleton. The junction is very strong. It connecting strongly two cells. This is a electron microphotogram of desmosome. Hemidesmosome, it's like half of desmosome inside one cell. Is a type of connection of cell with basal membrane or intercellular matrix. Adhesive belt or zonula adherens around the whole apical portion of epithelial cell. It also includes integral proteins of two cells with calcium ions between, and to these integral proteins connected this cytoskeleton. Adhesive strips similar but shorter between muscle cells. Occluding junctions, they are zonula occludens or tight junction, when two plasma membranes are between exciting cells, when ion canal of one cell connected to these ion canals of another cells, and when these connexons open, and the stream or beams of ions can go through very quickly from one cell to another cell. Cell cytoplasm consists of hyaloplasm or cytosol, organelles and inclusions. Organelles are constant structural components of cytoplasm. And there are general universal organelles available in all cells, like endoplasmic reticulum, mitochondria, Golgi Complex, lysosome, peroxisomes, ribosomes, cell center, cytoskeleton. And special organelles. They're available only in special cells, like cilia and microvilli in some epithelial cells, stereocilia, flagella in spermatozoa, basal folds in cells of renal tubules, for example, myofibrils in muscle cells, tonofibrils in epithelial cells. And another classification, and then organelles grouped on membranous and non-membranous organelles. And first organelle of general meaning, membranous organelle, we will discuss is endoplasmic reticulum. The network of canals and cisterns, the thickness between 0.2 and 1 micrometers, and their function, synthesis of complex organic substances and their transport to other parts of the cell. There are two types of endoplasmic reticulum: rough or granular, contains ribosomes on the surface, and it's responsible for protein biosynthesis for export outside cell to the needs of the whole body. And smooth, agranular endoplasmic reticulum contains no ribosomes. It provides synthesis of lipids and carbohydrates, as well as destruction of foreign substances, xenobiotics. And in some cells, it's also the store of calcium ions. Next, organelle of the general meaning and membranous organelle is Golgi Complex, or Golgi apparatus. Under light microscope, with special staining, it's visible as dots or lines, black in color. These are fragments of Golgi, for example, in nerve cells. This fragment visible quite well under electron microscope. It consists of package of flat cisterns up to 30. They concave, and the Golgi complex has two faces: cis face directed to cell nucleus, and trans face directed to cell membrane. From endoplasmic reticulum, substances inside small vesicles enter the growing cis phase, and then the substances slowly changes and release by the trans phase inside vacuoles. This is a scan electron microscopy of Golgi following freezing fraction of cell. And this is a reconstruction of Golgi complex in 3D. You see the flat cisterns, cis and trans phase, vesicles which enter the cis phase, and vacuoles which exit the transphase. This slide demonstrates the Golgi functions. Substances inside Golgi processing, complex proteins formation take place from carbohydrates and lipids combined with proteins, and where complex substances appear. And there is phosphorylation, removal of mannose, glycosylation, sulfatation, and phosphorylation of these compounds. Sorting for 3 groups of substances. Storage, accumulating before release from the cells. Packaging into membranes. And release when necessary. A release by secretion, a release and embedding inside cell membrane, endosomes and lysosomes formation. Endosomes, it's vesicle containing the enzymes, different enzymes, especially protease, which destroy organic compounds. First of all, proteins. Endosome may be early and late. In late endosomes, the pH gradually decreased, became acid, because membranes of endosomes contain protein pumps. They pump in hydrogen ions inside, and medium became acid. And lysosomes inside contain the pH less than 5. Lysosomes contain about 70 hydrolytic enzymes which digest all type of organic compounds, like protease digest proteins, lipase and phospholipase digest lipids, and nucleases digest nucleic acids, and so on. Which enzyme became active only in acid medium, so the membrane surrounding lysosomes contain protons pumps, which pump in hydrogen ions and decrease the pH of the content. There are 3 types of lysosomes. Primarily lysosomes, or Golgi hydrolase vesicles, separated from Golgi and contain inactive enzymes. And then pH decreased, they became active. It is the process of phagocytosis. It's called heterophagy. And then cell catch bacteria or foreign particle covered by cell membrane, and this structure called phagosome or heterophagosome. And if old defected organelles like mitochondria or fragment of endoplasmic reticulum covered by membrane, it called autophagosome. And primary lysosome fuse phagosome and produce secondary lysosome or phagolysosome. If many lysosomes are fragmented, it's multivesicular bodies. And the enzymes of lysosome digest foreign particles, bacteria, or organelles. And remnants, non-digested particles, release from the cell. If it's not possible, then they accumulated inside cell as a residual bodies, sometimes it called tertial lysosomes. For example, lipofuscin granules accumulated in old cells like nerve cells and cardiomyocytes. Another type of membranous organelles, peroxisomes. The vesicles containing enzymes oxidizing amino acids using oxygen. As a result, forming hydrogen peroxide or active compound, which further oxidizes the substances like lipids. It called lipid peroxidation. And surplus of hydrogen peroxide is destroyed by enzyme catalase. Sometimes peroxisomes contain crystal core. Mitochondria, also membranous organelles of general meaning, but it contains two membrane, outer and inner membrane, and inner membrane make folds, each called crysts, and between is matrix containing enzymes of Krebs cycle. It contains also DNA and small ribosomes and matrix RNA are responsible for biosynthesis about 10% of proteins of mitochondria. But other 90% of proteins of mitochondria coded by DNA of chromosomes of nucleus and synthesized in cytoplasm outside mitochondria by free ribosomes. In mitochondria, the oxidative phosphorylation take place. Organic substances like glucose or fatty acids oxidize inside mitochondria, and energy accumulated in adenosine triphosphate molecules here in oxysomes, in particles, in enzyme complexes, surface of crysts. The size of mitochondria between 0.5 and 2 microns. The lifespan up to 10 days, and the new ones appeared by division of the older mitochondria. Usually mitochondria has plate crysts, but in some cells the crysts are tubular and under electron microscope visible like vesicles inside mitochondria. Next, organelle of general meaning but non-membranous are ribosomes. They consist of small and large subunits, 10 and 15 nanometers in size. So ribosome has 25 nanometers in size. Ribosomes may be free, located in cytosol, or binding these membranes of endoplasmic reticulum, and they produce together rough endoplasmic reticulum. For ribosome produce proteins for the cell itself. Binding ribosomes produce proteins which enter the lumen of canals of endoplasmic reticulum and enter to Golgi Complex for export. And there are several millions of ribosomes in cell. Next organelle is cytoskeletal. Its complex organelle consists of numerous microtubules, microfilaments, and intermediate filaments. Microtubules consist of molecule of protein tubulin connected into chains, and chains produce tubule. These tubules have 25 nanometers in diameter. Microfilaments, 6 nanometers in diameter. It consists of actin globules, 2 chains of actin globules. Intermediate filaments, about 10 nanometers, and consist of fibrous proteins like keratin, for example, in epithelial cells. This is a very nice picture of cytoskeleton under fluorescent microscope. This is a nucleus in the middle of cells and numerous microtubules, the network in 3D of microtubules in cytoplasm. They really maintain the shape of cell, facilitate cell motility, forming of pseudopodia, cilia, flagella. They participate in endocytosis and exocytosis, this transport of substances and organelles inside cells. They participate in mitosis. They form in the achromatic or mitotic spindle, and participate also in intercellular communications. Finally, the cell center or centrosome, also non-membranous organelle of general meaning. It consists of 2 centrioles lie at right angle to each other, and every consists of 9 triplets of microtubules, provide like the wall of this cylinder. The length of every centriole about half of micron, and this is the electron microphotogram of cell center. One centriole and another transverse section and longitudinal section of centrioles in cell center. Centrioles produce all microtubules in the cell, or mitotic spindle, cilia, and flagella. Finally, there are 4 functional systems, apparatus of cell, like complexes of organelles which, under the control of nucleus, perform the important cell functions, like synthetic apparatus, including endoplasmic reticulum, ribosomes, Golgi Complex. Energy apparatus include mitochondria, apparatus of intracellular digestion and defense, include endosomes, lysosomes, and peroxisomes. Cytoskeleton include microtubules, microfilaments, intermediate filaments, and also cell center. Another big groups of the special cells, organelles, available in the special types of cells only. They may be as a projections from cell surface, microvilli, or a small finger-like extension of apical portion of some epithelial cells, for example, of intestine or kidney. They are responsible for absorption of organic substances, and they all together produce the striated or brush border. Cilia can move. They also extension of apical surface of some epithelial cells, but they contain the microtubules inside, 2 central pair and 9 peripheral pairs of microtubules responsible for their movement. In the basis of every cilia there is also basal body containing centriole with typical structure. You see cell cilia under scan electron microscopy like grass in the field. You see the transverse section of cilia under transmission electron microscopy, the pair of central and 9 pair of peripheral microtubules. And this is a longitudinal section of cilia with basal body. Stereocilia cannot move actively. They are elastic. If you move them, they return in the first position. Kinecilia, you see stereocilia without microtubules and one containing microtubules and can move. Other special organelles, for example, flagella in spermatozoa, has structure like cilia. Basal folds in renal tubules or myofibrils in skeletal muscle fibers. Inclusions, the temporary structural components of cytoplasm. They reflect the functional state of cell. So they can appear and disappear depend of the state of cell. There are 4 types of inclusions. Trophic inclusion as a store of food, protein inclusion, glycogen, carbohydrate inclusion, lipid inclusion, or pigment inclusions. This slide demonstrates the fat inclusions in a liver cell. You see the liver cell, nucleus, and black droplets of fat. And this is a glycogen inclusions in liver cells. Liver cell contain blue nucleus and pink granules of glycogen in cytoplasm. pigment inclusions in melanocytes. Contains a small granulus of pigment melanin, responsible for color of skin or hairs or eyes. Secretory inclusions, for example, in goblet cell. This is a nucleus, and in cytoplasm, the droplets of mucous secretion for release into the intestine lumen. Excretory inclusions is metabolic products for the removing from cell. These products not necessary for the body anymore, and the cells need to remove it. So, in conclusion, the cytoplasm composition, organelles and inclusions flated in cytosol or hyaloplasm. Cytosol, the true real solution of biopolymers, where organelles and inclusions are flated and suspended. In the medium, the organelles, inclusions and substances can move.